Okay, so there was our video. And if I can start this up again. Of course I can. Let's see. There we go. Chromosomal mutation. The position of genes can be altered in one of two main ways. So in transposition, the movement of genes from one part of the genome to another. So that's like where we have our gene 1, gene 2, gene 3, and one gene moves from one part of the strand of DNA into another part. Or we have chromosomal rearrangement, where it changes in position or number of large segments. So like crossing over, where the chromosomes that are the X's in the nucleus of the cell that are made up of all those genes, when it changes to a different leg of the chromosome, or it changes in the number of segments, so extra genes or missing genes. So let me give you a minute if you're writing. Okay. Here we have our changes in chromosome structure. So,
a chromosomal mutation is a substantial change in the structure of a chromosome
so
or
substantial, a big
One
time
way
change.
that this can occur is by a deletion or a duplication within one chromosome. In the chromosome shown here, the longer arm is labeled Q and the shorter arm is labeled P. Regions are designated with numbers. If a region is deleted, such as a large portion of the Q2 region shown here, then a deficiency occurs. There is now less genetic material in the mutated chromosome. A duplication occurs when an extra copy of a chromosomal region exists, such as the extra copy of regions Q2 and Q3 shown here. Both duplications and deletions can be created by abnormal crossing over during meiosis. An inversion occurs when a chromosome is broken in two places and reconnected so that a region is flipped from the normal order, such as the Q2 and Q3 regions shown here. Note that an inversion does not change the total amount of genetic material in the chromosome. Translocations involve two chromosomes. In a simple translocation, a single piece of one chromosome breaks off and is attached to another chromosome. In a reciprocal translocation, two chromosomes exchange pieces. Note that translocations do not change the total amount of genetic material. However, both inversions and translocations can lead to overall deficiencies and duplications of genetic material after meiosis.
Okay, so what they've basically said here is that if we stick with the same amount of genetic material, but it's just mixed up in different places, that's one thing. But if we delete whole pieces or we add in extra whole pieces, that's substantial. That's a really deleterious situation where something is going to be a major mutation. And one thing about this site, the McGraw-Hill Connect, is that if you look down here below each one of these videos, they have little practice problems. So I don't know if he showed this to you before, but you can go through and you can do practice questions when you're preparing for your test. When I prepare for tests myself, I always do practice tests because then I know what I don't need to study and what I do need to study. And that's, I think, a really good tip for studying. Okay, let's move past this one. Okay, what is biotechnology? So basically, we've talked about all of these different ways that that things can get moved around, or that amino acids can be changed, or entire proteins can be changed, and that when it happens naturally in nature, usually either we don't notice or it's a mutation that destroys the individual. But scientists have actually been able to harness this technology in order to engineer the genes themselves so that we can add in, delete, or transplant genes from one organism to another to alter the organisms in various ways. So this is kind of funny because it shows her with this turkey, and they genetically engineered it in order to have extra legs because that's everybody's favorite part. But that is essentially what is happening right now, is that we are genetically engineering our food sources so that they can either produce more meat, like if we genetically engineer cows, they can produce more beef, or they can be genetically engineered to produce more milk. Our turkeys and chickens can be genetically engineered to have these big giant breasts so that when farmers are raising them, they can get the most money out of having the largest turkey breasts or the largest chicken breasts. We can even alter organisms to do things that they never used to do before. Like if you've ever heard about golden rice. In China, I guess people were having vitamin A deficiencies, and so scientists were trying to figure out how do we solve these vitamin A deficiencies, and so they used bioengineering to change the rice plant so that it produced vitamin A, and that was called golden rice, and so the golden rice was more healthy, and it helped people in China who had vitamin A deficiencies. And so they've done that with our corn, they've done that with our soybeans, they've done that with our cotton. It's really quite amazing. And if we have time at the end, I will show you the TED Talk on how to build a dinosaur from a chicken using genetic engineering. It's really quite interesting. So let me. Okay, so the 5 tools of biotechnology. Basically, we have to have something to chop up the DNA from the donor species that exhibits a trait of interest. So, like, for example, so I'm diabetic. I've been diabetic for 30 years, and I take insulin. And when I first took insulin, the insulin used to be isolated from pig pancreases. So they would go out to the slaughterhouse and they would gather up the pig pancreases, and they would basically juice them in order to get the insulin out of them, and then I would get a little vial of insulin and I would take the insulin, and that would keep me alive. And pig pancreas insulin was great for the time, but it did cause some problems until they came up with genetic engineering, and they were able to take the gene to produce human insulin, and they were able to work it into bacteria DNA. So bacteria DNA and the human insulin gene, and these bacteria then began producing human insulin. And human insulin revolutionized diabetes because it was so much better for people than the pig pancreas insulin. So they were able to chop up the DNA inside of human cells, and they were able to work the insulin gene into bacterial cells and get the human insulin that they wanted. Another thing that we have to be able to do is amplify small samples of DNA into more useful quantities. So let's say you go to a crime scene and you find one little drop of blood. You can't do a test on just one little drop of blood. So what they would do is amplify the small samples of DNA. So take that one little drop of blood, make the DNA make lots more of itself, identical copies of itself. So amplifying, taking small samples and making it bigger into more useful quantities. The next step, inserting pieces of DNA into bacterial cells or viruses. So inserting the human insulin gene into bacterial cells, then growing these entire colonies of bacteria or viruses each containing some donor DNA. So taking the bacteria with the human insulin gene and making the bacteria replicate so that they have big, giant, massive amounts of the bacteria. And then identify the colony of bacteria or viruses that have the DNA for the trait of interest. So find the ones that are making the human insulin and isolate them. So not all of these tools are needed for every application. Some only utilize one of these tools, but these are the basic tools that we have of biotechnology. So the first one, which we'll be talking about a bit more in lab when we talk about DNA fingerprinting. The first step is to locate this gene of interest. So find the section of the donor species, the one that's going to give the bacteria the gene for it to produce. And then use restriction enzymes to target a particular base pair sequence on either side of the gene. So find the gene part that we want. So in this example, the restriction enzyme recognizes the sequence of A, T, C, G, A, T, and cuts between the A and the T. So these enzymes know exactly where to splice the DNA. And then the third step, the restriction enzyme binds to their target base pair sequence and cuts the strand of DNA. So notice that we've cut that DNA out, and so the gene of interest has now been separated from the donor, and it's ready to be put into the bacterial DNA. So that was one of the steps. Now notice it's a second step, but it doesn't happen in every one of the experiments that we do. We amplify, we make more of. Think of like if you have an amplifier for your sound system, it makes more of the sound. Amplify here, it makes more of the sets of DNA. So a solution containing an isolated segment of DNA is heated, and it's separated from the double-stranded DNA into two single strands. The enzyme DNA polymerase is added along with a large number of free nucleotides. So those little parts are floating around all around it, and it's heated up so that everything is moving around pretty quickly. And then the DNA polymerase adds those free complementary bases to each single strand. So that makes more of the DNA. So we start with one double strand of DNA and separate it into two single sides, the complementary sides. Then we add the base pairs that are all around it. We heat it up, and we begin getting the base pairs being added. And so we go from one strand of DNA to two strands of DNA to four strands of DNA to 8 strands of DNA, and so on until we have hundreds of millions of strands of DNA, and they're all identical. And it can be repeated over and over again until there is a billion identical copies of the target sequence. So then inserting the DNA using a plasmid, this one always reminds me of the insulin, where the bacteria makes the insulin. So targeted sequence of source DNA is isolated using restriction enzymes. So the source cell would be like my cell that makes human insulin. You take the human insulin gene of interest out of my cells. You put it into the plasmid of the bacterial cells. It is going to repeat, and it's going to make more and more and more of itself as the bacteria grow and divide into 2, and then the 2 divide into 4, and the 4 divide into 8, and the 8 into 16. So that means lots of my gene of interest have been made, and we have lots of copies of human insulin. One of the things that we can do when we grow lots and lots and lots of plasmids is that we can make up a gene library. So to create a gene library, a large amount of DNA is chopped up using the restriction enzymes. So if you look at step number 1, chop, 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 chop to get all those genes. And then we put those genes into plasmids, and that's the circular piece of DNA that's floating around inside of the bacteria. Put those plasmids inside of each of the different bacteria, and then allow the bacteria to multiply, and we have a whole library that has all of these different genes inside of it. And then we can identify the genes by using a DNA probe. So locate the gene of interest among the millions of clones in the gene library. The bacteria are washed with a chemical that breaks down the DNA, making it single-stranded. So we basically break apart all of those bacteria that we've created, and then break apart all of those strands of DNA, and then they put a radioactive probe inside of all those single strands of DNA, and it's washed over the single strands of DNA, and the probe is a short length of DNA that contains the sequence of bases complementary to the gene of interest. So imagine we have a whole bunch of pieces of DNA, and then we put in our DNA probe, and it's got the complementary set of DNA base pairs to the ones that we want. So it only hooks up to the DNA pieces that match those complementary base pairs. So it binds to it, it creates the double strand of DNA, and it's also radioactive, so we're able to easily identify it, pick out the ones that are radioactive, and we have exactly what it is that we want. So let me pause this one.